Well, thank you, Phil. It's a tremendous pleasure to be here for the Koch Symposium, and particularly on this topic of convergence. Phil asked 6 months ago or more if I would give a keynote talk on a personal perspective, personal reflections on convergence, rather than giving a talk with actual data, a talk with perspective. Because it's Phil, I said yes. But it also meant I had to go back and think about how in the world to frame such a thing. And so I have attempted to do so. It won't be identical to Phil's perspective, but it's quite aligned with Phil's perspective. And I'm just going to try to share just what it means to me, and particularly what it's meant to me within biomedicine, because I appreciate the theme of convergence is much broader. It affects food and energy and the environment and many, many other things. But I'm merely going to speak about what it means to me and what it has meant, what it means with respect to biomedicine. So what is convergence? I went to the MIT website and I found that there was a definition here, not unlike what Phil has told us there. Concisely, integration of engineering, physics, computation, and life sciences with profound benefits for medicine and health, energy, and environment. That's fundamentally about the process. And I do understand that the process is an important aspect of convergence, how it works. But if it's okay, I would like to talk about the question, which is relevant when you're talking about convergence. What are we converging to? Right? That's an important question. So I said I would talk about 30 years of convergence. It's backward looking to about 1985, but I didn't actually say which direction the 30 years would go, so I'll also think a little bit forward as well because we're not done converging by any means. So Phil gave his view of the 20th century, parsed in a certain way. And of course, we only really understand our histories when we have enough perspective on them. When we're living through them, it's very hard to figure out exactly what's going on. So I'm going to share my perspective, and I'll parse somewhat differently than Phil did, but touch on some of those same elements. But it's important, you'll see, for where I want to go. I read biomedicine, biology generally, but particularly biomedicine, is driven by 4 great intellectual revolutions which sort of squeak into the 20th century. 2 rather early ones. First, the revolution of biochemistry. Until the 1890s and really the early 1900s, biologists were still dealing with the idea of vitalism, that there were vital forces that could not be explained by the laws of chemistry and physics. And only with work of people like Buchner in purifying substances that could carry out glycolysis did we finally get the death of vitalism and the recognition that everything could in principle be explained. Almost at the same time, in the opening weeks of the 20th century, we get the genetics revolution, which is essentially the first time mathematical and abstract inference comes into biology. Here, of course, Mendel in 1865 is the first exponent of it, but it sinks like a stone into the water, and he's only rediscovered at the beginning of the 20th century and has intellectual descendants starting then. And so I'll put that there. And we have rules of heredity, and we have biochemistry, and they have nothing to say to each other because one is working from the organism down, the other from molecules up. In the middle of the 20th century, they meet because of the physicists who ask, how do we understand this abstract gene that the geneticists are working on in terms of physical substances like chemical matter? And we get Erwin Schrödinger's fabulous and very influential book, What is Life?, where he says, heredity, the gene must be an aperiodic crystal, whatever that means. And Max Delbruck makes that much more concrete by focusing and building a whole school that thinks about what is the gene. Of course, we now have molecular biology, but it's not actionable until recombinant DNA. Recombinant DNA represents a kind of engineering coming into biology for the first time. It's engineering using biology to directly purify and manipulate individual genes, which even as late as the late 1960s, people thought wasn't really going to be possible. And then it bursts upon us in the 1970s, and it is possible to do that. But that revolution still left us in a situation where if you wanted to find the gene for something, you had to know what the something was. Insulin, for example. You could find the gene for insulin or hemoglobin. But what you couldn't do was find the gene for something that you didn't know what the protein was. That meant you needed a prior hypothesis. It meant you focused on single genes, single proteins. And so what came next? Well, you could read the next part, as Phil did, as kind of genomics then comes along. But I would like to, in this context, view genomics as merely an early precursor of the kind of convergence that we're talking about. I would read instead what comes along starting in the mid-1980s and really only becomes apparent about now is something much more than genomics per se. It's a very radical view of biology in terms of completeness. That is, that biology really can be understood like we would understand an engineered object in terms of all of its components. I think genomics did much to provoke that, but I think that understanding is broader and broader and becoming broader and broader. To give you some sense of how radical that was, let me take you back to 1986, when there was the Cold Spring Harbor Symposium that took place in June of 1986 on the human genome, or it was the molecular biology of Homo sapiens. And there was the famous debate about should we sequence the human genome. And there was a meeting report written by the biology editor of Nature entitled The Proper Study of Mankind that appeared in the first week of July. Select quotes. Nobody seems to doubt that with improvements in technology, it can be done in perhaps a century. The question is whether it is worthwhile. As an information resource, the sequence of the human genome is an extremely doubtful asset. If modern biology is already stretched to interpret the sequences of known importance, such as the DMD and CGD genes, which were reported at that meeting, what possible use could be made of more sequences? It's a really wonderful article, and it says that there's an implicit assumption in biology that insight comes only from depth, not breadth, that there aren't unique things we learn from breadth. We begin to start thinking around this time in a more engineering mentality, in a more physical mentality, with the idea that biology may just be finite. Up to this point, biology was unbounded. There was always some new thing around the corner, some kinase nobody had ever thought of, some process you hadn't thought of. But at least at surprisingly many levels, we are beginning to find that the relevant components of biomedicine can be comprehensively discovered, comprehensively monitored, and systematically perturbed in a way much more natural to an engineer reverse engineering a system. And so what we need to focus on are comprehensive views and comprehensive tools. And that'll be the theme for much of what I talk about here. Now, let me note, I am not talking about systems biology, which goes much further than I'm prepared to go, which is, at least as sometimes described, say, by a colleague like Lee Hood, as writing the differential equations for all of life. I don't think we're anywhere near doing that. I'm not sure we can do that. But being able to systematically get, monitor, and perturb all of the relevant components and recognize them is something that I think is emerging. Moreover, there's an intellectual component in addition to this obtaining the pieces component, and that is that there are some things that we can best learn, indeed, many things we can only learn in hypothesis-free manners from the big picture. Around this time, there was still a great debate in biology, still is there today, about hypothesis-driven science as the only kind of science. Anything else was much less if it was not hypothesis-driven. And David Botstein, in the way that only David Botstein could, did a send-up of it by referring to it for some period as, oh, yes, that's hypothesis-limited science. And it was a really good point, because of course you were limited to what you could hypothesize, and it turned out life had been at this a lot longer than you were and was a lot smarter, and that hypothesis-free discovery was a very good thing. And if you had a very big picture, you could learn a lot from it. We've seen that, obviously we know that with regard to genomics, but what I think is it's still a radical idea, because what people have a tendency to do is admit that that was true for the last thing, but isn't going to be true for the next thing or the thing after that. And I think it's time to admit that this is kind of an idea that has legs. Now, I wrote the Completeness Revolution with an asterisk, because nothing in biology is ever complete. The human genome's still not complete. There's still a 0.5% running around we can't deal with. Everything I'll mention will always have some percent that we can't deal with. But when you reach the point where you're on the back nine, where you've got the vast majority of this stuff, we can declare that, for all practical purposes, a complete picture, even though, as a mathematician, that would offend me. As a biologist, I'm prepared to accept that, as an intellectual point of view, that kind of completeness is all right. So let me just sketch that arc briefly in the remainder of the talk. The driving force for this, the driving question, was to kill this idea of candidate genes. Through the 1960s, 70s, if you wanted to find a gene for a human disease, you had to start with a candidate. You know, perfectly fine for sickle cell to know you're looking for hemoglobin, but for cystic fibrosis, no idea what you're looking for, let alone for height and weight and predisposition to Alzheimer's and schizophrenia, and for cancer, not so obvious what you'd be looking for. Now, of course, Bob Weinberg and others could show that some cancer genes could be found functionally by demonstrating that they actually transformed cells, but there probably were a lot of other genes that caused cancer or played a critical role in cancer that we might not be able to find functionally because we didn't know the function we were looking for. And so a couple of simultaneous things. Renato Dobecco with an influential editorial in Science about cancer and the need to understand the genome, and David Botstein with this really seminal paper in 1980 about linkage mapping of genes even when you had no idea what the biochemical basis was, finding genes without a prior hypothesis. And by the end of the 1980s, there were in place very clear theoretical ideas about how to do mapping of simple Mendelian diseases in families, how to map the multiple genes that would contribute to common diseases in populations, how to find genes in cancer, but they all depended on the fact that you could wrangle the genome into submission. And the problem was that at 3 billion bases, the genome might as well have been infinite for all practical purposes, and the challenge was to render the genome finite and tractable. Thus led biologists to creating the second great periodic table, a kind of periodic table for biology. If the periodic table of Mendeleev gave us the chemical elements, the periodic table of biology gave us the genes, clearly critical, distinct elements that underlie biology. I will skip over the details of the Human Genome Project, other than to say it started modestly with just 400 marker genetic maps, but turned into a worldwide genetic project in 6 countries, many different labs, including here at MIT, which I'll proudly say, since we're here at MIT, was the largest of the contributors to that Human Genome Project. It got done multiple times. There were many celebrations announcing the completion of the Human Genome Project. They were all good celebrations. Of course, as I noted already, it's not actually completed because there remain little bits that we can't do. But again, for all practical purposes, we have a full sequence of the human genome. What's it good for? Well, from it, human DNA, human RNA, and human proteins are now essentially finite. We know the lists. Oh, I know, there'll still be RNAs we don't quite know, but the point is there's no doubt that all of that stuff will be mostly filled in, so we're at the 99% level on those things. Moreover, once we know them, we can ask intellectual questions. If a protein just got a phosphate group added by a kinase, there's no choice. It's got to be by one of the known kinases because there are no other kinases than the known kinases. Similarly for many other functions. And that means you can never say it's due to a kinase we've never met. That's a really deep and important intellectual thing. In addition, signatures. we can recognize a bit of protein by a fragment in a mass spec because we see its amino acid, a little bit of amino acid sequence. We know there's no other protein that has that sequence, or there is one that does. Same is true for RNA. So it brings this into a kind of engineering realm. So at that point, there were very hot debates, including amongst people involved in the Human Genome Project, of were we now done and could we go back to business as usual, or was this paradigm more generally applicable? Were there other things? I remember many hot debates, including with Maynard Olson, that no, this was the end of the line for this point of view. We should go back to. It was interesting. There was clearly one place where the point of view of completeness was very important, and that was for finding those disease genes. So the next thing that got done toward this goal of completeness was the study of human genetic variation. And it became clear that common genetic variation in the human population is finite. There's just a limited amount of it, and it can be recognized by signatures. And this was seminal work by Mark Daly when he was actually a student in my own laboratory, where he showed that the genome had an elegant haplotype structure. There were a limited number of haplotypes in any region of the genome, and you could find them and understand them. And you could use a few markers to recognize all of them. And that quickly led to the characterization of essentially all common genetic variation in the human population. Anybody in this audience, if I pull a variable base from your genome, the chance is 99.8% it's already in the databases. And you can look them up in databases like EXAC and Nomad, maintained by Daniel MacArthur, for example. It's a very useful thing to have. So the next question was, what about rare genetic variations that aren't common? Well, here, they're not finite, I have to admit, but they are tractable because the cost of sequencing genomes dropped by 3 million-fold, and you can just look for them as needed. So that folks back to the question of hypothesis-free gene discovery. Could we discover the genes for rare Mendelian diseases, common polygenic disorders, and cancer without a prior hypothesis of what they were doing? Could it be done in a systematic kind of way? Well, for Mendelian diseases, it turned out to be a piece of cake. They're finite, they're tractable. Victor McCusick's famous old catalog of Mendelian inheritance in man listed thousands of diseases, and as the tools got better and better, it turned out that most of them could be found very, very easily. Many more could be found with a little more work. There's more than 4,000 known today, and that's the majority of the ones we actually know. There's still more, but nobody doubts at this point that this is a finite problem, with finite having an asterisk that there'll always be some things we don't know, but it's a small proportion. With regard to polygenic diseases, harder. Here you're taking populations and looking for multiple genes that contribute, and primarily for the important ones. And to make a long story short here, using these methods of common genetic variation, it went from an incredibly slow process of one disease gene a year in all diseases combined across the entire world to, as tools began to develop, 4, 8, then in 2007, as the tools were really there, a lot. And then by today, 30,000 significant associations across hundreds of diseases and traits. And we now have lots and lots of regions of the genome that we know contribute, and people are poring over them to find the genes. But interesting things, and this is very much a big picture question that would not have been part of an old biology, people like Hilary Finucane take the millions of sub-significant genetic variants and show that they have patterns that can teach us about the disease. By taking the not yet significant, in fact, a whole list of genetic variants, she can figure out which tissues are critically involved in a disease. And as we get more knowledge about cell types, which specific cell types are involved. You can learn from all of that information in a kind of mathematical, computational way, things that you could never imagine learning at the laboratory bench. And so it's led to people having very concrete lists now. Inflammatory bowel disease, 200 genes, entirely new processes people didn't know before were involved. Autophagy, similar stories can be told for early myocardial infarction, schizophrenia, obesity, and all here I've chosen things from the collected MIT and Harvard community. Of course, cancer. Cancer, a very similar story. Systematic approaches have led to the discovery of more than 400 cancer genes so far, and Gady Getz will, I assume, I know because I talked to him yesterday, talk about a bunch of this, so I will say very little other than to say systematic approaches didn't just find the receptor tyrosine kinase pathways that we knew, they also led to many things that we did not know in advance. They pointed, before experiment did, to chromatin regulators and remodelers in cancer, tissue-specific transcription factors, RNA splicing factors. HLA started coming up, which of course we understand in terms now of escape from the immune system. And so I'm sure you'll hear very much more from Gady about that. But that brings us to this point of saying we've got a genome, RNA, DNA, RNA protein. We've got human genetic variation. We can begin to get fairly comprehensive views of the contributors, at least the pathways for disease. We won't get every one of these genes because many have very little effect, but the pathways seem to be emerging. Around this time, or mid-2000s, efforts began to focus on regulatory elements. All right, genes, great, 21,400 or something like that, protein coding genes. How many regulators in the genome? More comprehensive approaches have been developed over the years now for being able to identify those by evolutionary conservation across species, by comprehensive epigenomic modification mapping, ChIP-seq and other techniques, that we now have extraordinarily detailed maps of where these putative regulatory features are. 3D folding patterns of the genome by techniques like Hi-C, and in the past 3 or 4 years, particularly around MIT, people, including Jesse Engreitz at the Broad and MIT and Harvard, have been making the beginnings of a case that we may be able to build a comprehensive map of enhancer-promoter connections. We're not there, but it raises this question of, can that be rendered finite and tractable too? And then going further, signatures of cellular processes. How finite and tractable can they be? Todd Golub and many collaborators involved in this broad community of cancer geneticists and genomicists at MIT and Harvard and the Broad have been involved in asking questions like, how well can we capture the cellular signatures associated with drug perturbation, gene perturbation, using RNAi originally? And by collecting large numbers of them, they have more than 2 million profiles right now, can we infer when two drugs have the same target, when this drug affects this gene? Can we begin to recognize a finite collection of fundamental underlying signatures of cellular processes? And for this purpose, and when we see a new mutation in a cancer gene, if we put it into a cell, can we tell by the cell's response? Typically here, this is being done by gene expression responses. Can we tell whether that's functional or non-functional? Even if we don't know what the function is, can we tell whether it's benign, because there's no meaningful response, versus that these mutations trigger a similar response in a known pathway? It's by no means done, but it's a very, very interesting idea, and there are more and more hits coming out of it to the point that you can imagine that, life has a finite number of processes. Maybe that'll be 30,000 processes, but 30,000 is not as big a number as it used to be. And then, of course, when we think of comprehensive tools, one cannot but think about programmable genome targeting, particularly CRISPR. CRISPR is a GPS. It's a genome positioning system. People talk about CRISPR as genome editing. That's one thing you can do with it. What CRISPR is a genome positioning system. It has a way to, in a programmable fashion, send whatever you want, any protein, or actually any RNA too, to any location you want in the genome by just writing its address on the top of the envelope, and it goes there through an appropriate guide RNA. That's an extraordinarily general, comprehensive tool. And then through work by Ophir Shalom in Feng Zhang's lab and Tim Wong and David Sabatini in my lab, the development of whole genome CRISPR screens to be able to knock out every gene in the genome and systematically find every gene that contributes to any cellular process that you can monitor by selection. So for example, cancer or growth of cancer cells, HIV replication, drug resistance reporters. Not all processes yet, but we'll come back to it. So it means that being able to produce comprehensive lists of all genes meaningfully involved in a process can get done in a more systematic way. Then, that was 2013, by 2014 began to emerge the notion that human cells should go on the list of things that we should regard as finite and tractable. That just as genes are kind of the atomic elements of heredity, cells are the atomic elements of the body, and that there's a need for a third great periodic table to join the table of chemical elements and the human genes, a complete periodic table of the human cells. And there is a project, and Aviv Regev here at MIT and the Broad has been, I think, the greatest driving force in this project, to create an entire human cell atlas over the coming years. It was inconceivable only a few years ago to do such a thing because you couldn't analyze individual cells on their own for their gene expression patterns. You could only analyze averages. But the technologies for processing single cells, sorting them, preparing libraries, barcoding them, mixing them, sequencing them, analyzing them by computer has led from a dozen and a half cells in an early paper not very long ago to millions and millions of cells being analyzed just here at MIT, Broad, Harvard alone. And so it raises the questions, how far are we going to be able to go toward this notion of the full periodic table? Classifying the types of cells, the states of cells, the transitions, the locations, interactions, and lineages directly. And the reason why it's powerful is because you can think about it in terms of mathematics, computing, and engineering. Every cell has coordinates. If we just think about gene expression, you could add other things, but I'll just think about gene expression. 20,000 coordinates corresponding to the 20,000 genes describes a cell. That's a very rich description. If a cell is really doing something very different, some of its coordinates will be very different. And so you should be able to identify cell types by classifying cells as regions of space, and development by trajectories in space, and states of cells. And you should even be able to classify tissue in terms of the kinds of generative models that generate the mixtures of types of cells and tissues and infer interactions. And Aviv and her colleagues have done a tremendous amount of work. And I won't talk about the implications for cancer because she is on this program and we'll talk about this very shortly. And you will hear about that. But I note that this interacts with everything I said before. Before, when I said Hilary Finucane was trying to figure out which cells in the body, which tissues in the body were involved in any given gene disease by comparing the lists of genetic hits to expression profiles of different tissues, that was done on averages. This is now doable on single cells, and we're beginning to see already that the single cell analysis will tell us not just it involves the immune system, but it involves this kind of cell in the immune system with great precision. Similarly, if we cross together two technologies, the cell atlas and CRISPR genome screening, before I said you could only do CRISPR genome screening when you could select for a given phenotype, growth or activation of a reporter gene. Well, not no more. You can now take single cell analysis, throw in a CRISPR library, and in every individual cell read out the full perturbation on gene expression. And in that way, begin to study any process you want in an extraordinary amount of detail. And the questions will become, as in engineering, how from such data do we reverse engineer the circuitry? It's a kind of breathtaking thing that has swept over this period. But there's still more to go. New frontiers. There are many new frontiers. The challenge I would leave, particularly the scientists at the Koch, but across all of MIT and Harvard, is an increasing need to focus on programmable therapeutics. I begin to wonder whether 100 years from now, people will look back on the late 20th century as this barbaric time when we had to find a specific molecule to do each job rather than to write code to do the job. I don't know what I mean exactly by that, but I put a little icon here of a programming book teaching C++ and saying we need D++, where D is DNA in some way. We have already some remarkable things to look at. DNA-directed chemical synthesis, where people like David Liu can force any reaction to happen by bringing into proximity, to infinite proximity, two molecules attached to complementary pieces of DNA and driving chemistry. We have DNA-encoded chemical libraries, where collections, many small molecules, each attached to DNA, can be easily screened to determine which are binders to your target of interest by simply seeing who binds and cleaving off the DNA tags and reading what happened. If you built the molecules and you've tagged the molecules, you can do that. Rapid directed evolution efforts in many labs. Again, David Liu's lab is an example of this. With the synthetic biology work going on here at MIT with Chris Voigt, for example, and Ron Weiss, the idea of building truly therapeutic circuitry. We're still far from D++, but they are working toward D++. And then, of course, we need programmable delivery. Not all diseases occur in the liver, which is where most things will go on their own. We need to be able to program delivery to any cell type, modular nanoparticles, the kind of work that Sangeeta Bhatia and Bob Langer think about. Or, if we can't perfectly program, we could also have programmable activation. When something gets to, it might get to 5 cell types, but in the right cell type, it gets activated. And we need to think about this with the generality that we think about everything else. I note at the bottom that the President's Council of Advisors on Science and Technology that I served on for 8 years thought all this was particularly relevant to the nation with regard to biothreats. We need to have development cycles for new drugs that are measured in months, not a decade, in order to be able to respond to the kinds of threats that may emerge. And the last thing we need is, the other thing we need a comprehensive view of is healthcare. That the healthcare system right now could not properly be characterized as a learning system. It is a system where information is balkanized, locked up, unable to be integrated. And there are efforts afoot to bring this same comprehensive view to the healthcare system through patient-partnered research programs like the Count Me In efforts that Nick Wogley and Corey Painter are leading here in Boston, and things like the Precision Medicine Initiative underway in Washington. We need to reach a world where all that information can be integrated. So this completeness revolution, or completeness star revolution, because the little asterisk it signifies, as I always signify, we will never be perfectly complete about any of these things, but it doesn't matter. Close is very good in biology. Many of these things are either in hand or in prospect, and that's really exciting. Now, of course, none of these are the goal. The goal is actually medicine, at least in my view, but this is the foundation. These are the tools. We can't forget that even if we did all these things, it doesn't complete the work of solving disease. That work may never be done. But the tools to do it, the understandings to do it, the comprehensive views and comprehensive tools are coming into view. And I think that, to me, is the answer to Phil's email of what my perspective over the last 30 years of convergence has been. This is what convergence is about. It's not all that convergence is about, but this is one of the key things it is about. I want to close by saying that my thoughts about this, which have formed over 30 years and are still subject to ongoing revision, have been informed by many, many people. I put up a list of people in alphabetical order who I have learned a tremendous amount from and had long discussions with over the course of years in shaping my own views, and more broadly, the whole communities of the Broad and the Koch and all of MIT and Harvard. And with that, I will stop. Thank Phil again for this hard invitation, and congratulate us all on having what will be an exciting symposium on convergence and cancer. Thank you.